Welcome back to Season 2 of That's So Second Millennium, the Catholic Science podcast, where we look forward to the new synthesis in the new millennium between faith, philosophy, and science. Welcome back to episode 66 of That's So Second Millennium. This is the continuation of our interview with Maureen Condic. We start with a fairly technical but very intriguing discussion of stem cells, the different kinds of stem cells, different words, including one that Maureen has felt the need to coin herself in order to clear up certain confusions about the nature of different kinds of stem cells. We talk about early embryology, some fascinating stuff that I managed never to hear in high school biology. I don't know about the rest of you all. And then Bill asks a very intriguing question. How can this wonderful entity that we've been discussing, the early embryo, the blastocyst for that matter, which in some sense might really be a, quote, clump of cells, but boy, it's a fascinating and beautiful, in the sense of what it can do, clump of cells. Bill asks how this entity can be dismissed as a clump of cells, and that allows us to transition to an interesting discussion about the motivations of scientists, especially biological scientists, and the problem Maureen sees in her field of people who desire to, you might say, make the fly. I made that fly. I, changed its genetics. I made that fly. And the fascinating and disturbing sense in which they almost might see God as a competitor in the art of creation rather than rejoicing in the creation that allows them to do this tinkering and experimentation and exploration. And then we conclude with a discussion of the difficulty that she had actually writing a book with her brother in terms of simply the language barrier that exists between scientists today and philosophers because of, and we went all the way back, if you were to go back to our first few episodes, we talked about the divorce between science and philosophy, and Maureen bears witness to that in her own field, and the importance, in this book that I really want to pick up that she wrote with her brother, we'll link it in the liner notes, that spends a considerable amount of time, and she thinks the best part of the book, simply bridging this terminological and conceptual gap. So this is a fascinating episode, and we will have one more. This is going to be followed up by a third and final episode next week, and then we will move on to interviews of the other speakers at the conference. So without further ado, here is Maureen Condic. We've talked about these terms, and I was doing a little bit of reading, not as much as I would like to, of some of your work before putting this together. So we've used the word, I believe, pluripotent already, and you have used the words totipotent and plenipotent in print as well. What if we could break down what the definition of just those three terms are? They're sort of
I did coin a new term, which I did with great trepidation, but I did it precisely to try to get around an incredible misunderstanding in the public and a misuse of terminology by the scientific literature.
Yeah.
So, totipotent is a term that means having all powers, able to do everything.
Yeah.
And the scientific literature uses that term in two distinct ways. Mm-hmm. The first one is the ability to make literally all of the cell types that come from that one cell embryo. So this would include all of the cells of the embryonic organs of the placenta and embryonic membranes, as well as all of the tissues and cell types that exist in your mature body. So if you can do that, you are totipotent. You are the kind of cell that has the ability to make everything.
So the zygote is totipotent.
The zygote is
the gold standard.
The zygote is totipotent, but so are many stem cells in this limited sense, in the ability to make all of the cell types of the body. Mm-hmm. So in mouse, for example, stem cells taken from embryos are pretty much restricted to making just the cells of the mature body. So they don't make the cells of the placenta, for example.
Right. They've already.
But human embryonic stem cells actually have the capability of making everything. So they're totipotent in that simple sense, the ability to produce all of the cell types that are normal human cell types. Mm-hmm. The stricter sense of totipotent is the term as it would apply to a one-celled embryo. So a cell that has not only the ability make all of the cell types of the body, but
in
to
fact,
organize
it's going
them
to, and to
in
organize
a
them.
coherent, rational sequence that allows for, I can't say rational, but in a coherent sequence that allows for the ordered production of increasingly mature states of human development.
Right.
So if you have that kind of totipotency, so if I take stem cells, for example, and I aggregate them all together, they can and do produce all the cells of the body in a chaotic mass like a tumor.
Yeah.
But an embryo will produce all of the cells of the body in an ordered developmental sequence that progressively moves towards more mature states of the human form.
Yeah.
So that kind of totipotency is the kind of totipotency that really is all powerful, can do everything. Pluripotent, in contrast, refers to cells like I just described, the mouse embryonic stem cells. So cells that have the capability of producing all of the cell types found in the mature body, but not all of the cell types in human development. So the cells of the placenta and embryonic structures that are transient. So pluripotent is able to make all the cells of the body. Totipotent, in the strict sense, is to be an embryo, is to make all the cells of the body and also organize them in a coherent body plan. And we needed something to replace that simpler sense of totipotent, which is confusing when people use it, because people will read that and say, oh, they've made embryos. And what they've really done is make a tumor that has this broader developmental capability of producing all of the cells that are possible to be produced. So that would be plenipotent.
That's fascinating. That is a whole world of, when I early on decided to go the opposite direction of biology, it's like, oh yeah, I'm leaving that behind, aren't I? So that does bring up the question, which I believe you've addressed at length in different places. At what point because of course there's the phenomenon of twinning, of identical twinning, at what point does the zygote, at how many divisions down does a human, as opposed to a mouse or a turtle or a chick or whatever, how many divisions down does it lose the ability to be fully totipotent in that sense?
Yeah, this is a really, really important question. In fact, I have a book in press at the University of Notre Dame Press called Untangling Twinning
There you go.
that addresses both the philosophical and the biological issues associated with twinning in great detail. So the short answer to your question is proving a cell is totipotent. It takes a very strict scientific test. You need to isolate that cell from all other contributing sources of information, and then you assess what its capabilities are independent of any other cell type within the embryo. Now, the hard thing about that experiment is that cells don't like to be isolated, and it's very hard to do it in a manner that you don't get the negative finding of the cell dies, or it doesn't do anything, it just sits there. So when that happens, you don't really know what its potency is, because the only way you can assess it is if you get a positive finding. Right. So with that as a caveat to looking at the strict question of totipotency, the ability to not only produce but to organize all of the cell types in the body, there appear to be species-specific differences in how long that capability is preserved. So in mice, I think the weight of the data is that the first two cells produced from the zygote, if separated, can go on to produce independent little twin mice. But after that, you lose the ability to do that. In some species, for example, cattle, for some reason, seem to preserve totipotency up to, say, the 4-cell stage. Pretty routinely. So people clone cattle fairly frequently by blastomere separation, by separation of the early first 4 cells of the embryo. And each of them can routinely go on to make a little baby cow.
It's progressed to the calf stage.
Right. There's one unreplicated report in a particular species of pigs that asserted, that I would just call this an assertion because it's never been shown by anyone else, that in that species you can preserve totipotency up to the 8-cell stage. So each of the first 8 cells, if separated, could potentially produce an embryo. But I think in the vast majority of mammals, the 2-cell stage is about as far as you can go. I think cattle are really the exception. I think this one report in pigs is probably not correct.
Happens sometimes.
It does happen. or it was an almost finding that nobody's been able to replicate since then.
Right.
But that's not the only way that twinning can happen.
Mm-hmm. Then there's fraternal twinning.
There's fraternal twinning where you have two independent embryos and they just so happen to coexist in the uterus at the same time.
They have to be womb mates.
But there's also the capacity to develop as an entire embryo, as a whole, unified whole, can exist in a multicellular embryo as a whole. No one cell, if you took it out, could restart the whole process of development all over again.
Right.
But the entity as a whole has that capacity. So, and there's very good experimental evidence and some evidence from fertility clinic work that splitting an embryo much later, an embryo that contains hundreds of cells, will cause each of the halves to heal and go on in development to make twins, identical twins.
Yeah.
So that kind of splitting, that isn't separation of the earliest cells, but an actual division of the embryo in half, that can occur up to about 14 days of human development. Okay.
So we have some evidence that happens naturally as well as, or at least we have no reason to believe it doesn't happen both at the two-cell stage as well as.
We have good reason to believe that it never happens at the two-cell stage. Although experimentally we can do this.
Okay. But all human, in the wild, identical twins.
Identical twins. I'm going to argue, I argue in the book that I think the vast majority, if not all, identical twinning in humans in the wild, so to speak, in natural situations occurs at the blastocyst stage, which is a stage where you have many hundreds of cells in the embryo. Yeah. It looks kind of like a hollow ball.
Yeah, because it has both the wall of the placenta that's forming as well as
the inner cells that are going to go on to produce the postnatal body.
Yeah.
So all of that early development happens inside a protein coat known as the zona pellucida. It's a very rubbery, tough coat that protects the embryo and the egg. The egg when ovulated, and then the early embryo as it's traveling down through the fallopian tubes to the uterus. But in order for it to implant, you have to escape from this rubbery coat because the outer cells.
And that rubbery coat is also generated by the embryo.
The rubbery coat is actually generated by the egg as it's developing. So it's a structure surrounding the egg.
Oh, is that different from the corpus luteum?
Yes.
Okay. Yeah, there's so many things. So many things.
Yes. The corpus luteum is a structure within the ovary. Part of the organization surrounding the maturing egg called a follicle.
Right.
And so the cells of the follicle that remain in the ovary after ovulation are the corpus luteum.
Yeah.
But the zona pellucida is this proteinaceous coat. It's very simple. It consists of 3 proteins, creatively called zona pellucida 1, zona pellucida 2, and zona pellucida 3. But they make a nice, tough, protective coat for the egg. The sperm actually have to penetrate through the zona in order to get to the surface of the egg to have fertilization happen. And then all of early development happens within the zona. But in order for the embryo to implant, the outer cells actually of the embryo actually mediate implantation. And they have to escape from this covering, or they can't interact with the uterus.
Yeah.
So this happens through a process called hatching. And it actually, I have some great movies of this. It's just unbelievable. It really does look like the embryo secretes some enzymes that weaken the zona A1 region. It's usually a small tear, and then this entire structure will squeeze out of that little eggshell.
Yeah.
And people have observed twinning happening at that exact point.
That would be a point where you would expect that to happen.
Two little bubbles instead of one little bubble.
Yeah.
And so long as each of them inherits some of the cells in the inner cell mass, you get identical twins.
But are there times when it divides in a less fortunate manner, and then you simply, unfortunately,
one that just has some cells missing, but they're not sufficiently large enough group of cells for them to carry on the development on their own and regenerate.
Oh, that gets complicated really fast.
But I think part of the confusion about twinning is the belief that twinning happens at the 2-cell stage. And we can do this in the laboratory. People do it routinely. But it has never actually been observed.
Okay.
despite the fact that in IVF situations we have daily observations of embryos. Many hundreds of thousands of embryos have been watched over many years, and no one has ever reported this happening.
Nobody has seen that happen. Okay.
Even though the actual rate of twinning in IVF situations is highly elevated compared to natural.
It's what? It's higher?
It's quite a bit higher.
Quite a bit higher, yeah. So in that situation, which no one should be exposed to, but that's the world we live in. So that whole, and I forget the name of it already, but that protein coat is present as well, and that's growing in the dish or what have you, and so that has to swell, and then at some point that gets implanted, and this whole tearing process could also happen. I mean, there would be a more or less normal rate of that style of twinning. Well, I mean, that would be you're saying it's higher.
Yeah,
and that's when it would happen.
Yeah, and some of the thinking about this from animal models is that the longer period of time you have the embryos in culture, the tougher that protein coat gets. Yeah. And so the likelihood that you're going to have a really constricted aperture through which the whole embryo has to escape would potentially increase the likelihood of twinning.
Yeah, the adventures that we all have to go through without, before we have any chance to remember any of it.
It's fascinating. I've often been asked how, my experience as a scientist has given me a better appreciation for humans and for human life. And, honestly, it wasn't until I was pregnant and I really started thinking about what I knew about embryology in a practical sense and in a personal sense that I became completely flabbergasted that anybody ever survives to maturity.
Right? Right?
Just, how can this ever work?
How does this? It's all got to go just right.
Exactly. And there's so many variables and they're all having to happen exactly at the same time, in exactly the same way. And oh my gosh. Yeah. It really is. It does give you a sense of awe.
Yeah,
and
without question.
The miraculous nature of human development that we ever get out here more or less intact with 5 fingers and 5 toes.
Right. Yeah.
Given that whole realm of awe and wonderment and amazing unlimited potential for different things to happen at that stage of life, I have to just think as a Catholic layperson that, in the Catholic imagination, it seems so viscerally sad and disturbing that this wonderland, in a sense, is so easily dismissed as a clump of cells.
Yeah. No, there's that. I was thinking the side of we invade it and manipulate it to our own ends so readily, but that side of it too. Yeah.
Both of them are disturbing, and also I find that attitude among my colleagues. I don't want to say disingenuous because I honestly think they don't think about it, but it is a distortion of reality and of a reality that they should know better.
Yeah, and you spoke early on about that. What compelled you early on was the big picture question, and what you've been discussing here is a lot of big picture, big possibility kinds of questions. And presumably you're not alone among these biologists. They too were drawn in by big questions, big picture at some point in their early lives. And yet it falls by the wayside, and that too is viscerally sad. It seems to be.
I don't know if anyone's ever done a sort of sociology study of what draws people to science, because I'm not sure I would endorse the concept that most scientists are drawn to it by big picture questions. Really? At least not in biology. Well, I think in biology, the vast majority of people who are drawn to it are people who, took their mother's toaster apart when they were 8 years old and confident that they could put it back together, except for that last spring
or something.
It didn't quite fit.
Or the thing that crossed it and shorted it and sort of got the Pop-Tart on fire.
You might have had a toaster experience in your past.
It wasn't a toaster per se, but some things of that nature.
Okay, well, so I think biologists in particular are not people who spend a lot of time contemplating the big pictures. They're not like cosmologists or physicists or maybe geologists who have this huge.
There are different branches. Yeah, I mean, the people who go into oil and gas are, they have an end and a means to that end, as opposed to the people who study the history of the planet as a whole. Or planets now, multiple planets as a whole. Those are fairly big picture questions.
Yeah, so I think that in my experience with biology across many different organisms and different orientations on more or less pertaining to medicine or to basic science, the really unifying thing is the tinkering mentality. I want to be able to take things apart. I want to put them back together in cool ways. I just want to see if I can do it. If I can take that toaster apart and put it back together. And I love to tell stories. This is how I order my mind, I think.
We're doing a podcast, so this is exactly when you should exercise that. That's excellent.
So I had a postdoctoral advisor who was a fabulous fly geneticist at Berkeley. Really just the kind of mind that you could just stand in awe of. He would lecture, and pose some biological question. Have you ever wondered how gene X interacts with gene Y? How would you test that?
Right.
And then he would just, off the top of his head, describe these incredibly complicated genetic processes. Everybody's frantically taking notes, and I'd go home and an hour later work it out. I guess that actually would do what he said it would do. But he was so experienced.
The logic of...
So logical, he could just do these things off the top of his head. But and amazingly, as many scientists are, a confident individual. And I remember more than once, after going through one of these very, very complicated genetic processes to test a particular idea, he would pause dramatically, with his hand on his hip, and his other hand on his chin, and say quite smugly, "So, I made that fly." I made that fly. And the fascinating thing is that attitude. No, I didn't test the hypothesis.
No.
I didn't discover the information. No, I had an idea in my head, and I manifested it in reality.
Yeah.
I made the fly.
Right.
That attitude, it's tinkering writ large. It's the ability to have an idea and to make it happen. Not to discover what's in nature, but to make the fly yourself.
Yes, it's not discovery. It's a creatorship. It's would it be hyperbolic to call it a kind of God complex?
Because it sure sounds like that. Yeah, I mean, that's the whole...
Well, but that kind of power, that it's so intoxicating. I mean, people who love the experimental side of science, who, biologists I know, myself included, would wake up in the middle of the night and look at the clock and go, okay, the experiment's been running for 4.5 hours. If I ran into the lab right now, it would be gone enough so I could tell if it worked or not. And they would get on their bicycles, drive to the lab at 3 a.m. because when it works, it's such an incredibly powerful feeling of, you had an idea, you made that fly, you made it happen. In reality, it isn't, and it almost is completely stripped of any sense of discovery. There's almost no wonderment about it. It was that you were clever enough to make it happen.
It's sad to lose that sense of wonder.
Yeah. Really. Well, it's sad and sort of contrary to the scientific enterprise. We consult reality and see what it has to say. Well, I mean, and of course, there's the whole, there's both a philosophic and a spiritual side of, you were able to make this fly because there were already flies.
Yes.
Everything from the fact that there are things rather than nothing, all the way down through the existence of laws of physics that allows complex phenomena like this to happen, to the construction of a genome where there's already all of these pieces in place, and you took some bricks out and slid them in different places and something cool happened. And that's great. But
you did not make the fly. It is, and one of the reasons I tell the story is because the obvious absurdity of that claim. I would often push back on him. Oh, so what chemicals did you start with? When you made the fly, I would love to do that myself sometime.
Yeah, exactly. Yeah.
I'd make a really big one, so I could fly to work out.
There you go. Exactly. Yeah, that's right. I'm gonna go down. I'm gonna buy some coal, and I'm gonna purify it, and I'm gonna get the carbon and hydrogen out of it, and I'm gonna make a fly. Let's start from there.
Right. And it's odd because again, they're so poor. There's such a complete lack of any kind of philosophical training for the vast majority of scientists. They just don't talk these things.
Yeah.
But it struck me in your litany of everything from the Big Bang all the way up through to the existence of a genome.
Yeah.
I would say right before that, you still have to have the existence of the soul. You have to have a substantial form for any living thing that is kind of the gravitational forces towards which all of those biological processes are working, and that hold them together as a coherent thing. So that when you take out a few genes, sometimes you kill the fly, but you rarely cause it to break down into carbon. That result has never been obtained.
It hasn't gone all the way back that far. Yeah, that is. That's fascinating to me, and I don't know enough Aristotelian and Thomistic philosophy to do this yet. Maybe I'll be crazy enough to go off in that direction. But old Aristotle and his form and matter idea.
Yeah.
It seems to me there are a lot of ways it works better with modern science than it did when you were trying to say, well, it's a table. What's the form of a table? Well, it's kind of has to be sort of like this. Whereas what's the form of an electron?
Right.
Well, that's pretty precise.
Yeah.
What's the form of Drosophila melanogaster? There's the DNA, the genome is a fairly precise set of rules. It's not that hard, but then that would be only part of the form.
Well, it's a very so prior to this book that's coming out on Twinning, I wrote a book with my brother Samuel, who is a philosopher, and to my knowledge, this is one of the few books that has really attempted to bring to bear onto the question of anything, in our case, the embryo, a rigorous philosophical analysis and a rigorous biological analysis. And the interesting backstory to this book was that the hardest part of writing it were the 3 or 4 years where we just had to fight endlessly over terminology. Like, what do you even mean when you say this?
Right? Yeah. And that's one of the most generous things you can do is to put the result of that argument out there for other people not to have to have the whole argument.
We were both convicted that we were talking about the same world. And if you're talking about the same world, you have to develop a common vocabulary that allows you to talk about it intelligently and a common shared set of understandings.
Yeah.
But as a result, when we had finally worked through mostly an education on my part, learning biology is relatively simple, but wrapping your head around hylomorphism
said the biologist.
is much more challenging.
Yeah.
I think that the amount of information you have to acquire to wrap your head around biology is enormous, but the complexity of the ideas they're all fairly trivial.
Yeah.
Overall,
a very large number of relatively small inferences to get to where you're going.
Whereas trying to understand what is the form of a Drosophila is a much harder thing to do. But I would say the second biggest fight was once we had gotten to the point where I could barely hold on to all of this and think about it intelligently. I'd insisted, we have to have a chapter in the book that addresses this from the perspective of for people like me, people who never really thought about it this way before. Yeah. And to my brother, that was just everybody knows this. Of course, it's obvious. Why do we need to talk about what hylomorphism is?
The dangers of specialization. Yes.
So the first chapter in that book is, I think, a great. It reflects me, the ignorant scientist, trying to. This is my understanding. Endorsed by somebody who has a much more natural sense of the relationships between the various components, and it writing that chapter really convinced me that intrinsically all scientists actually are Aristotelian. They look at it that way. They're Thomists. They have this understanding. We just don't know they have it because you can't really do science without an understanding of the fact that the subject of your experiment can change and remain the same thing that it was, despite change. Yeah. Because if we didn't have that, we would never be able to do an experiment. We'd never even be able to interpret our results because every incidence would be its own independent thing.
Right. Yeah. What is the title of that book?
That book is called Human Embryo, Human Being, a Scientific and Philosophic Analysis.
That's right.
is that important that very few books like that have been written that attempt to synthesize the philosophical and philosophical
I think a lot of people try to do it, but they do it very poorly because they don't go through that 3-year process of really working to try to understand or work with another person of sufficient competency. So you have a lot of philosophers who dabble in biology and come up with some grand unifying theory that is completely
doesn't really contact the truth on the ground.
Well, it's completely uncoupled from the facts. Except for sort of the high school level facts that maybe are 40 years out of date and overly simplified. And then you have a lot of scientists who get old, get tired of writing grants, and suddenly imagine themselves to be philosophers. Hey, I wrote Kant once back in high school. And,
right.
Picked up a few modern,
I think Bertrand Russell or someone.
That's right. That's so I'm confident, and now I'm going to speculate away about the meaning of the universe because I'm old and crotchety, and I know I'm smarter than most everybody else I know, and so I should be allowed to do that. But of course, they do it very, very naively and completely uncoupled from any kind of philosophical rigor or because it's hard. It's hard. I certainly couldn't have done this on my own. I mean, we had a true collaboration, and it was I think very certainly for me a very productive one, and I hope a useful one for everybody else.
Interesting. I mean, and to take that and I mean that is the thing is. There is like you said, there is so much philosophy out there that sort of attempts to, and it's sort of glibly, of course, from my own specialty, I can look at things, are you really defining that? From the perspective of geology and chemistry, that's not necessarily an adequate treatment of that. And that's, and then of course, the other side of, the scientists who even bother to convince themselves they need to write something about it. Gotten on that opinion of themselves, and that seems to me completely. I mean, that would be what's required to build that bridge. It was what we would have to do now to capitalize on the philosophy and the science that we have. The way that it was, again, you can't replicate it, and it would be much more complicated. Thousands of people instead of the several dozen that probably gave us the medieval synthesis in the 13th century. But what work could be, I mean, to me, even if you are, even if you're just laboring away trying to, okay, how am I going to apply this to the question of what is a tectonic plate? or crystals or minerals. What should we define as a mineral species? Where should we draw those lines? Should we have all these names? Maybe even just that. But that would still be, I mean, so I use this example, from earth science. If you've seen 3 tin mines in Cornwall, you think you know how tin deposits work. And then you might go to Spain and you're like, as it turns out, cassiterite can crystallize in this completely different environment. That's interesting. I better do some more work and see if there's a common. And then go to Indonesia. And by the time you've got 30 tin mines, you might not be as surprised when you go and see tin mine number 31 and say, okay, this actually is at least like existing patterns. And that I don't know that there's a need to go deep and there's a need to go broad. There's also kind of a need to be at that 45 degree angle in between. And I think that's really where there's stuff lacking in terms of, I think the philosophical ideas would get a lot more rigorous if we expose them to enough different environments and demanded an actual, what would work with the facts on the ground and this science and this science and this science. And you would start to see patterns of things that I think we're missing.
I agree.
It's an idea.
And I think we have this throughout the scientific enterprise. As someone mentioned earlier, the need to, the demands of science to falsify your own hypothesis is a really, really tough thing to wrap your head around. And in practical fact, people tend to shy away from looking at evidence that could disqualify their hypothesis.
Yeah.
And that even applies to things that they've never really formulated that good hypothesis on. Everybody knows.
Right.
And so let's not actually look into the literature and find out if that's true or not, because I'm comfortable with everybody knows. Because it supports, whatever vision of the world I want to have and allows me to just assume that there's no alternative explanation.
That's So Second Millennium is brought to you by me, Paul Giesting, and by Bill Schmidt. Find more of Bill's work at onward.net. If you enjoyed this episode, please share it with a friend. You can email a link to this episode at thatsosecondmillennium.net, share the post for this episode from our Facebook page, or you can use your podcast app's built-in sharing feature.